Physician researchers at City of Hope have developed an innovative approach to overcome the challenges of resistance in chimeric antigen receptor CAR T-cell therapy, a groundbreaking cancer treatment. Published in Nature Biomedical Engineering, their new method allows T-cells to carry multiple features, addressing various tumor escape mechanisms simultaneously. This advancement aims to enhance the effectiveness of CAR T-cell therapy, especially for patients who experience resistance. CAR T-cell therapy works by reprogramming a patient's own immune cells, known as T-cells, to recognize and attack cancer cells. These T-cells are engineered to produce a receptor, called a CAR, that targets specific antigens found on the surface of cancer cells. Once infused back into the patient, these engineered T-cells bind to the targeted tumor cells, destroying them. However, some tumors develop resistance to this therapy by hiding or reducing the expression of the targeted antigen, making the cancer cells invisible to the immune system. The researchers at City of Hope, led by Dr. Scott E. James, have tackled this challenge by developing a system that allows for encoding multiple features into T-cells. This dual-vector approach increases the genetic storage capacity of T-cells, enabling them to target up to 4 different antigens simultaneously, compared to the typical approach of targeting just 1 or 2. Dr. James explains that this advancement is akin to increasing a computer's storage capacity with a flash drive. Using a single vector, there's a limit to how much genetic information can be added to a T-cell. By using 2 vectors, researchers can double the genetic storage capacity, allowing them to encode more complex programs in the T-cells. The researchers demonstrated the success of this method by engineering T-cells that targeted multiple antigens and included switch receptors that can convert negative signals from cancer cells into positive signals, reducing T-cell exhaustion, a major barrier to effective therapy. Collaborating with institutions like Memorial Sloan Kettering Cancer Center and the University of Pennsylvania, the team has shown that this new dual-vector system significantly boosts the anti-tumor activity of T-cells. The modified T-cells were able to proliferate more effectively, live longer, and resist immune suppression by the tumor. This innovative approach, called zip sorting, is a powerful tool that could help researchers build and test more effective cellular therapies. The researchers are currently testing this method in mouse models, but their long-term goal is to optimize it for use in human cells. Their early success in mice is a promising step toward overcoming the multiple resistance strategies that tumors often employ. In addition to targeting multiple antigens and combating T-cell exhaustion, the zip sorting method could be used to introduce other enhancements, such as transcription factors to improve T-cell proliferation or safety switches to control T-cell activity. Dr. James expresses his excitement about the potential of this method, noting that the ability to engineer T-cells that can resist multiple immune evasion strategies is a major breakthrough. It was surprising that we could add so many features to a T-cell and still maintain its activity in a tumor microenvironment that would typically suppress it, he said. This innovation holds great promise for improving the efficacy and longevity of CAR T-cell therapies, and the research team hopes to continue exploring additional ways to enhance their approach. By overcoming the limitations of current CAR T therapies, zip sorting could pave the way for more successful and durable cancer treatments in the future.